So, despite the echo, that is not me. And what we're going to look at today, your challenge is how are we going to look at things in the water column in a monitoring sense. I'm going to use marine renewable energy, but that's really just a venue. Think of any ocean observing or environmental monitoring. And before I launch into it, I also want to acknowledge the students that have been partaking in this program. We've run 3 and now we're working on a 4th master student in this evolution, as well as programming assistance from Ross at the end there. So hopefully I acknowledge those as we go through, but I want to make sure that I get them up front. Okay, so I said environmental monitoring. What does that mean? Well, from an acoustic perspective, we'd use a bit of jargon. We'd say pelagic Nekton monitoring at marine renewable sites. But for those who are not familiar with acoustics, and Phil's here, we might say stuff in the water column, because that's really actually a term we commonly use to describe things we don't know. So we're going to look at stuff in the water column, and how do we track it? How do we figure out what it is? To give you just a little bit of context, an environmental monitoring plan is actually part of a license application in the US and part of your consenting process here in the UK. But to this date, there are no standards, no procedures, or even any best practices for any sort of monitoring. And this applies to many industries, many applications. And for marine renewables specifically, each license for each site is negotiated separately over what can be extended periods in the order of a couple of years. So if these standard methods existed, it would streamline and reduce costs of both permitting, consenting. It would enable comparison among sites, which we can't really do right now. And I hope lead the industry to best practices. So that's my goal, to try and convince you that it's possible and maybe give you some tools to be able to do it. So what's the primary objective? We want to measure change in monitored variables. And when I'm going to define measure specifically or explicitly, being the ability to detect, quantify, and forecast change. We're going to have 3 components to this. First, we want to match our sensors to our objectives or to our variables. And given the increasing amount of remote sensors that are being used, ultimately we'd like to reduce data volumes and eliminate what's become known as the data mortgage. We ultimately want to define, detect, model, and forecast change. And to be acceptable to the industry, we'd also like to enable operational monitoring. And I'll go through these, and these are specifically color-coded to kind of give you a sense of where we are as we go along the path. Just to familiarize everyone here with what happens at a marine renewable project, you go through what I'm calling the life cycle, where you start out with a potential site selection. You're going through your permit or consenting process. You've got your installation. You finally get to your operation, and then ultimately you may have a decommissioning of any devices or any obstructions that are in the water. So in these 3 steps, I'll break it down to first characterization, looking at specific metrics and data reduction. Then my English teacher will be rolling in his grave, but we're going to talk about spatial representativeness, a nest monster. From that, though, it enables you to design a monitoring network. Now that we've got our network set, we can look at what we'll use as threshold to monitor change. And I'm going to adopt extreme value analysis, and we'll look at site specificity to see if it is generically possible. And we'll get more to the monitoring, where we're going to change detection or for change and look at detection, modeling, and forecasting. And then introduce you to a bit of software that we're developing, which enables real-time device-animal interaction and the looking at biomass flux. There's a set of terms used in ecosystem monitoring that's specifically been adopted for marine renewables, where we start with some sort of a stressor, and we'll just call that generically some human intervention. We have a receptor, and we'll have our generic fish, but it can be your beast of familiarity or favorite. Then you may have 1 or more indicators, which look at characters of that receptor. You may or may not be able to measure those indicators directly, so you may have a set of metrics. And what hasn't been done, and what I'd like to introduce at this time, is a modeling of the process which enables that detection and the forecasting of change. So that's the orientation we'll use in trying to figure out our tools. In a project that was done in Monterey Bay a few years ago, I put an echo sounder down at about 875 meters. on smooth plain looking up. And what you see here is 2.5 days of vertical migrations of plankton and mesopelagic fish heading to the surface at night, dispersing and then descending and setting up layers. A universal pattern in any deep water. You also see individual fish down at the bottom for those who haven't seen an echogram. And just a quirk, this blank spot is due to a rodent chewing through a 52-kilometer long cable, and I thought you'd appreciate this, which took down the entire system. Even though this thing was at 900 meters, it was a rodent on land. We've developed a set of metrics, partly extracted from landscape ecology, partly from the acoustics world, to extract more and be used as descriptors for our raw acoustic data. The top two are very typical, and they talk about they indicate the relative density or the absolute density of animals in the water. These are calculated over the entire range, the 875 meters, but we can get more than that. We'll look at center of mass, where the weighted mass, mean of the biomass is in the water column, the variance around that center, the percent of the water column occupied, how evenly is distributed. We can look at the patchiness, and then ultimately we used also an image analysis technique to count the number of layers. And that was used as a setup now, because we can take those metrics, use them as covariates, use them as response variables, but they actually give you a sense more of some of the structure and function of the ecosystem you're looking at. And in this case, we can look at the density measures, or the center of mass is how the biomass is distributed, or the structure of the distribution, and how it's dispersed, or how that dispersion changes, and how aggregated it is, more at the function. So we can get ecological meaning or interpretation based on these metrics derived directly from the collection from the sensor. To address the data volume reduction, I've got an example here. This is a multi-beam sonar, and what you see going across the screen is actually a dolphin coming across, and you'll see a turbidity crowd, and he'll march his way across. And we'll see this again near the end. And if you were to monitor that animal using this system at a 20-meter range at a 10 hertz ping rate, 50 kilohertz of data sampling, 1 minute of data is going to give you, 1 minute of acquisition is going to give you a gig of data. You run that for 30 days, you end up with 97 gigs. You run this set of metrics over the same set of sampling conditions, and you'll end up with 2 megabytes of data. So if you can store the raw data, but in terms of monitoring, if you can actually use the metrics, you've reduced your data volumes by 4 orders of magnitude. And what that means is you can do your calculations in real time or near real time. Very useful for trying to respond to events as they happen. And the nice thing about the metrics is the values don't increase as you increase the resolution of your data or the number of beams within a particular sensor. So I'm going to say that's a way to reduce the data volume, and we can move on from that one. The case study or example that I'm going to use is in Admiralty Inlet on the west coast of North America, outside of Seattle and Puget Sound. And it's the major entrance to Puget Sound, a major waterway on the west coast. And we set up in the spring or May or June in 2011 surface surveys where we fished and had surface acoustics and had a bottom package right off Admiralty Head, which was the proposed site for 2 open hydro turbines, which have been put on hold right now in this package using a Biosonics 120 echo sounder. An important part here is the mobile and stationary surveys were conducted at the same time, so we did have simultaneous data. Okay, having got the simultaneous data, that enables us to think about how much space does a point in time represent. We've got spatially indexed data from the surface survey. We've got temporally indexed data from the echo sounder on the bottom. The preoccupation in marine renewables for the monitoring has essentially focused on device monitoring. And that's really the concern about animal-device interactions. Sorry. As an ecologist, I had the timidity to ask what is the effect of the devices as opposed to the effect on the devices, but that's another story. When we get to commercial scale operations, we're covering tens of kilometers. Think of the big wind farms that you can see in the surface, and they're talking about tidal turbine farms. So now we need the ability to characterize the site or the domain. And so I want to look toward that transition of domain monitoring. As a practical application, how many monitoring packages are needed to characterize an entire site? And you can optimize your monitoring power to cost. And of course, if you're running a business, you want the biggest bang for your buck. So we had a look at 6 different ways to translate spatial variability to temporal variability. 3 by direct comparison, 1 in looking at scaling the mean using autocorrelation, and a couple ways looking at the variance where we're scaling the variance looking at equivalent scales or theoretical spectra. When looking at ways to compare means, you're also able to estimate a linear or areal interpretation error. So it gives you a sense of your uncertainty or your measurements. I'm not going to go into the details, we've got lots of ground to cover, but if I translate that to more of a practical example, on the left are the 6 methods, the 4 for the mean, the 2 for the variance. In taking each one of those techniques, this is your scalar to give you your representative range from a point to a linear dimension. And you can see that they range over 2-3 orders of magnitude. Translate that to a density of devices per kilometer. You also get a range of values. And if you were to take a $75,000, and you can pound, euro, whichever currency is up at the moment, to give you a sense of what that would cost for your monitoring program. This last column is the estimated cost per square kilometer to get 100% coverage in any domain at that density. So you can see it ranges quite a bit, and it's worth having a look at. But at least now we have that scaling factor, so we can realistically translate what an environmental monitoring program might actually cost. What that also enables us to do is to follow through a thought process getting towards designing a network for monitoring, whether it be marine renewables or anything else. So if you have a quantity of interest, you choose a metric that is indicative of that quantity. Are you interested in the metric property of, say, the mean or the variance? What is your objective? Are you trying to detect change? Are you just interested in the spatial distribution? Are you interested in spatial temporal equivalence or quantifying the spatial variance? Those are the 6 methods listed as shown in the previous slide, and then you can estimate the error dimension for 4 of those in either areal or linear context. By putting these pieces together, it enables you, before you go out the door, to start think about how would you design a network of instrument packages to look at a domain or a site. So you need to choose your monitoring variable or variables. You need to choose your metrics and instrumentation to monitor those variables. Are you interested in the mean or variance as a property? And then what's your objective? Are you trying to detect change? Are you trying to get information about the spatial distribution? Or are you trying to estimate some sort of an error or precision within a population estimate? Having made those choices, you can go out and acquire your baseline spatial and temporal data. So I'm advocating both a mobile and a stationary survey. And with that data, you can now calculate your representative ranges. You can potentially calculate your interpolation error and end up practically with a density of sensors. So now we've got a monitoring network. Our next challenge is how much change constitutes real change. And I'm just going to deal with it in this sense of statistical change. We won't talk about biological change. I'm much interested in having that discussion later on about statistical versus biological significance. But looking around, we've adopted extreme value analysis, which is often used in hydrology and engineering. And the classic example is the 100-year flood. How tall do we make our dike? How strong do we make our bridge to withstand some given risk? And for extreme value analysis, or EVA, the beauty of that, it enables you to model the rarer values in distribution tails. Those extreme events are usually considered the ones that have most significance, but rarely seen, which is a good thing in most cases. And given a couple of ways to do it, there's a peaks over threshold method, which defines all extreme values as those above a threshold. So it enables you to quantitatively or objectively fit a threshold based on an empirical distribution of your data outcomes, and fits a generalized Pareto distribution, a GPT, to these extreme values. So here's an example using the density metric of the Admiralty Inlet data, where this is just plots of the density function of densities. Here is our objectively determined threshold, and then there's this fit of this generalized Pareto distribution. You can see that it enables you to model things that you haven't seen. We'll use this in a bit. The other tool that comes with this approach then is you can also calculate what are called return levels. And these are average periods of extreme values, and we've added in a way to calculate Bayesian confidence intervals. So this is essentially the probability curve of observing those extreme events at any given period of interest. So here's where you can build for your 10-year flood or your 100-year flood and look at how typically your intervals are much narrower here, because here's where your data density is, and you're getting off to a much less populated data density to estimate those uncertainty levels. Okay, extreme value analysis. There's a tool we think is reasonable. Can we put that toward standardized monitoring, or is standardized monitoring possible? In the marine renewable energy world, sites are chosen typically for their physical attributes. Tidal turbines, you want good flows. You want high surface waves. There's really no guarantee that your biological attributes are going to be similar at those. And if they are not, then each site is unique. You need to set up a monitoring program that is unique, but there's a possibility that they may be similar. So we were very fortunate to, and this is where Beth Scott comes in, and Benjamin Williamson, where they had a similar acoustic data set connected at EMEC out in the Orkney Islands at the Fall of Horne. They're in red. Admiralty Inlet is in blue. And we've got 4 of the 6 metrics here: the density metrics, the center of mass, the variance around the center of mass, and the patchiness scale. And these are the time series that were actually very similar in the time of year, very similar conditions, collected independently, sampled at the same density. Surprisingly, quite similar, just in the characteristics, the biological characteristics. I'm not making distinctions of species. We're looking at whole water column. Theirs was about 35 meters, ours was 55 meters. It was slightly encouraging. And then when we went to see if we could apply the same technique to start to talk about thresholds and therefore talk about change, the upper 2 panels represent the Admiralty Inlet EVA, or extreme value analysis, with the fit and the return periods. And underneath is the same plot done up for the Fall of Horne. When we first saw the values of the thresholds between the 2 sites, we thought for sure there might be something wrong, because they're completely selected independently. Now you might think that the similar physical conditions would lead to similar biological conditions, but these were very similar in a threshold, which would be a change in density. And you can also see that the fits of the GPDs, the generalized Pareto distributions, do differ and also result in different return period curves, but that's also a function of the data density as you get out in those higher values. What I take away from this, though, is that these sites were biologically similar enough to warrant the potential for standard monitoring methods, and that was very, very encouraging. Because up until this point, remember, every site is negotiated back and forth between the agency and the proposer or the developer to figure out what to monitor, how to monitor, what tools to use, and this can take a period of over 2 years. Well, let's go back to our roadmap, so to speak. And now we've gone through identifying receptors, talked about indicators leading to metrics, but we haven't touched modeling yet, and that's the next step. The challenge here, we want to detect, model, and forecast change. So we've used this approach, and I want to identify kind of the steps that we took before showing you some results. Dove into the literature and wanted to see what was used broadly for monitoring. Having done that, we also looked at the tools that were used to identify, or what models were used to identify those changes. And we ended up choosing 11 models out of 3 kind of regression groups: generalized regression models, time series models, and non-parametric models. We think of it as having representatives of these groups, as well as using the most common tools that have been used in previous studies. So it's a combination of known and unknown. The next step in this approach was to fit to baseline data. We used the Admiralty Inlet data set. And to show you that one, here were two metrics, the density metric and the patchiness metric. And the point here is these are representative of what we feel are our general data classes, where we have normally distributed data and we have non-parametric or non-normal distributed. And you can see in the patchiness index where we have kind of two states of very low background patchiness and then these high spikes where things become very discrete patches in there. So we fit, and this is an example, an ARIMA model, arithmetic moving average model, fit to both of those. And that gives us characterization of our baseline conditions. Then what we did is we thought about what are reasonable things that represent change. And in the case of marine renewables, we came up with a set of stressors, being noise, the presence of a device, the operation of a device, and a chemical spill, and then a parallel set of 5 what we felt were reasonable biological responses to those changes or those stressors. A step change, a linear change, a nonlinear change, some sort of periodic change, which is the only one that varied the variance, and then some combination of a step and a nonlinear change. Having done our simulated change, then it was time to evaluate the models. And just to give you a sense how Hannah became the second greatest high-power computer user on the university for a while, we generated 19,000 data sets. From those data sets, we chose sampled over close to 2.5 million for the power analysis. And then to do the fit and forecast metrics, which I'll tell you about in a minute, that was another 41,000 samples. So it's, thank goodness we have computers. Not to go into the detail of this, but this is just to kind of give you a sense of the 3 groups of regression models that were used, color-coded, and the individual models that were used within the groups and how they might be representative of those groups. The rest of the table is looking at the form, whether they're parametric or non-parametric models. If they're looking explicitly at portions, if they partition the variance, does it include an autocorrelation factor, and how's the error distribution? So you could search for your favorite model or just get a sense of the variety of tools that are potentially available. Most studies to date have used 1, maybe 2 to compare, but there has never been a systematic comparison in the marine renewables for sure, and in many other fields, just looking at a wide range of these models. So how do we actually evaluate them? We use 3 criteria. First, we were interested in looking at their ability to detect change. For that, we used a power analysis, or you might call it a pseudo-power analysis, gone along the lines of the intervention analysis literature, where you have an indicator variable from representing before or after the intervention, so that you can compare the statistic to see if the model actually detected change, and through 1,000 simulated data sets we can actually estimate the power. The root mean squared error, you're familiar, R-squared metric, we use to look at model fit and forecast accuracy. So as a smaller value is better in this case. And finally, to evaluate the ability to forecast data by looking at 90% and then the other 10%, we used a MACE statistic, or a mean absolute scaled error. And in this case, a MACE value less than 1 indicates a greater forecasting ability than a random walk. We want to really make this an even playing field for all of the models evaluated. These metrics also enabled us to do that so that we could have a level playing field and yet also allow each model to optimize its strengths, because they're designed for different purposes. When you go through all those simulations and all of the fitting, all the forecasting, and looking at all of the results for those 3 metrics, this is the tree that we came up with. And it's not a quantitative tree, it's a schematic to indicate, given your receptor, fish, generic fish, where you're looking at normal or non-normal data, those attributes, abundance, distribution, diversity, all typically had normal distributions, where the behavior had a non-normal distribution in the outcomes. Where you're interested in looking at the mean or the variance of those attributes. And where you're interested in quantifying, detecting, or forecasting change, those are the recommended models based on our evaluation statistics. What we're trying to do is to provide a logical approach to the choice of your tool, given the conditions under which they were evaluated. But you can see a single or possibly a pair of models that are used depending on the objective. Just to look at one of those branches and to connect it back to our roadmap a little bit, we're back to here, and that was this left-hand branch of that schematic where we have looking specifically at abundance, which is a normally distributed variable. We're interested in looking at the mean and to quantify change, and the recommendation then would be use a moving average regression combined with a GAM model. So now we've got a set of tools, a way to quantify what constitutes change, a toolbox which enables us to choose the appropriate tool to model change. How do we bring that more into a practical setting? And think about a person sitting at a desk with a bank of monitors. The devices are there. We have our sensor packages that are looking at them. And we've got two concerns. Any species with special status, we typically have special rules for. Whether it be a salmon, whether it be a marine mammal, and depending where you live, it may differ. So collisions or interactions between animals and devices are of importance. And if you're thinking about a population or an ecosystem, typically you think bigger than an individual. And so now I'm back to stuff, where how would the interactions or the movements of these be affected and potentially impact populations? And that's where I think is the logical next step analytically. What we've done, though, is develop a set of middleware, and right now the first version is called NIMS, where we take live feeds from sensors, and in this case this is a picture of the multi-beam, the M3 head that I showed you that dolphin video from, and a almost current technology, the EK60, which is a standard echo sounder, where we intercept the raw data that come up and have a feed to our display, which we then can run a tracking algorithm to look to see as animals are approaching a device, because we're assuming that the package is projecting out from a single device or in the vicinity of a device. And at the same time, we have parallel processing going on where we're calculating those metrics in real time. The advantage of having the metrics is, given that we've established thresholds using the EVA, we can impart thresholds to single or combinations of those metrics. And any time those values exceed the metrics, the red light starts spinning, the big Auga horn goes off, and the operator can hit the big red button to shut things down. We can register it as an event, therefore we can start recording raw data, metric values, and then also produce standard reports that may go back to the regulatory agency, to the operator, for further analysis or documentation at least of some sort of impact. That impact may require operational modifications or even mitigation, depending on how the monitoring plan has been set up. So there are two things that it focuses on: the device-animal interactions and the biomass flux. So I can show you the user interface, and there is our dolphin, our porpoise dolphin, going through the cloud. There is a turbidity cloud, and for the most part, in the same step size, the metrics are being calculated in real time. And remember, this data is coming in as actually 10 frames per second, and we're keeping up in this calculation, so it's not bad. We have isolation that you can look at particular values as you're being monitored in real time. We're thinking about the user. We can capture images at any time, just as recording events, and then we also have a whole control panel where we would set up all your parameters, input your thresholds, and affect your data streams and your display. We see this potentially also enabling a conversation between regulators and developers. So in using the metrics, they can look at what constitutes events and how things might be monitored. So to summarize this approach that I'm advocating, we start off with a site characterization, which includes both a surface survey and bottom sampling. I feel that that indicator metric approach can be used with any data stream. Now, there is an assumption about the monitoring network. It assumes stationary packages, and these marine renewal projects are typically thought of as the order of about 20, anywhere between 20 and 30 years. So the thought there is that a stationary package on the bottom is going to be a lot cheaper than sending people out on boats and regular surveys monthly or weekly. Often a lot of effort and more reliable data sometimes. The extreme value analysis does provide an objective determination of statistical thresholds, and I already stated that there should be some thought about do they actually mean biological thresholds. The model chosen to detect, quantify, or forecast change really depends on the indicator or metric property of interest. But together, I think that some sort of standardized and best practice monitoring is possible and actually advantageous in this field and others. And that real-time monitoring with a user interface is possible, which facilitates these regulatory standards, event triggers, and recording and reporting. Collectively, I've shown you an approach, some example hardware, analytics, and software, hopefully to combine an efficient and effective domain monitoring system. And I'm really interested in your thoughts and responses, and that's all I have for today. Thanks very much.
Thanks, John. Any questions?
I wonder if you can give us some example, what actually can be discovered by using this standardized method. I don't know if this is already in place somewhere and you are currently monitoring some sites using this standardized method, and what we can learn from actually using standardized method rather than kind of, as you said, this site-specific method. Because I understand the main question is, you know, how devices affect fish before and after.
Okay.
Yes. So what we can learn from kind of globalization if each site is specific?
I think there are two primary advantages. The question was, what can you avoid, what can you gain by using some sort of standard methods? And I think I would focus on two things. First, it really is an arduous process to set up these monitoring plans in the industry. It takes several years. The baseline studies can take literally millions of pounds or euros or dollars to conduct. And there is no set of guidelines that anybody's working with to determine that you're actually going to measure what is significant. What is considered significant depends on what is known about the system, the local experts, and often the one who has a loud voice that is advocating either for or against the commercially most important species. So there's one side. And the other side, think of it from a research perspective. Because different techniques, different sensors, different analytics are used at each individual site, there really is no way to compare results to see if the impacts, the potential impacts of any type of device or an individual device at one setting is equivalent, similar, or different at any other device. So if you're thinking about truly a global industry in this case, there really is no way to do a wide aperture data analysis.
Is any results already available, or this is just in the process of collecting data in terms of research application of the standardized method?
To my knowledge, there really is no wide comparison. The ability for us to compare Admiralty Inlet to the Fall of Warness is the first that I'm aware of. So it was pleasantly surprising that things came out as even as they did. Yes?
How does this work in a far field scenario, like, rather than looking at the local effects, but kind of far field effects?
Okay, that's a great question. And will you define far field for me, because there's different uses of that term?
I don't know, like, you know, beyond the kind of
vicinity?
Yeah.
Okay. So the question was, how can hope to paraphrase, how can we take results from a set of sensors in a particular spot and scale it up, so to speak? I think that's a great question and a next challenge, and it's actually just been the last week that I think I've got an approach that could be used. With this preoccupation of device-animal interactions, it's been a very myopic aperture, mainly because of the endangered species or the special status species. I hesitate, but one individual suffering injury or mortality may be a small or a significant part of a population. But how do we get population effects? And I think that is the next challenge. We don't have too many subsurface marine renewable programs yet. Maygen is the first one coming online. But if we're interested in how these may impact populations, then we have to look at what constitutes stressors, to use that same terminology, on a population. If we think about it in terms of changes in biomass, then we can think of demographics, births and deaths. We can think of kinematics, active and passive locomotion. So are they going with the flow, which is possible in these tidal turbine sites? Are we actively swimming? Demographics, kinematics, and somatic growth. And I think that gives you a pretty reasonable set of stressors or processes that you can use to evaluate what impacts your population of interest. Having done that, then you can take those same processes and say, what indicators do I need that is actually going to enable me to monitor what's important? And the only other twist in there is that we've got to operate over a range of spatial and temporal scales, potentially, in different life history strategies. But I think it's doable with some work. At least it gives me a way to think about it.
I was wondering, how were you able to get about 2 million samples? What duration of time?
Okay, so that question was about the simulation for the change scenarios. We use the Admiralty data set as our baseline data. We put in functions to represent each one of those 5 different scenarios of change, and then generated those 19,000 simulated data sets where we had the baseline and then tacked on the simulated data sets. That's what we use to sample for each one of our evaluation metrics for each model in each form. So we were continuously going back to that pool of 19,000 data sets to then use to tabulate or evaluate the different metrics. So we kept drawing from the same pool with replacement.
Yep. So yeah, I'm just wondering, so you said earlier that's an arduous task on an individual.
I'm going to come over a bit closer because I can't quite hear.
You said that for individual sites it's quite an arduous task to put any sort of plans in place and data monitoring, basically. So I presume what you're proposing cuts down time on the data collection.
Cuts down time.
For data collection, for monitoring. Is that what takes the most time, or is it the negotiation of creating and applying a plan? Because if
okay. Well, so the question was, will the use of these packages, correct me if I'm wrong, will the use of these packages enable a more efficient or cut down on the time of sampling to get towards an end product of a monitoring program? Some of those things are done in serial, and some of them are done in parallel. You pick a site, you start to do an initial survey to get your baseline characterization in order to put together your application for a permit or consenting. And I may be out of step here on this side of the pond, but then it's up to the regulators, in the case here for the biology, it'd be your fisheries agencies, to say, okay, these are the things that we think you should do for mammals, these are the things we should do for seabirds, for fish. So the acquisition of the data, except for the fact that you've got remote sensors going all the time, and if they're cabled, you're getting that data in real time, that gives you more volume. I don't know if it reduces the total time, but I sure hope that it increases the efficiency and the effectiveness of what comes out in the monitoring plan, because you've got a comprehensive set of not only the data, but of the way to analyze the data and to speak a common language between those that are collecting the data and those telling you how you're supposed to be collecting the data.
Any other questions? So I've got one, John. You see the different models that you had for the different types of data, essentially. In terms of your performance, you identified those that were best for each case.
Okay.
And there was a variety of them. I just wondered if, given the complexity of putting the whole system together and understanding of how to run those rather complex models, was there one that actually did it well enough in all cases that you would say, well, if you did, you know, these are all the best cases for each case, but overall,
yeah.
You could you use one that would do it all pretty well?
We sure would have liked to, but to keep it objective,
Yeah.
given those evaluation metrics that we use, those three, there was no clear winner, so to speak. It was an even head-to-head competition among them all, but the way that the models were structured, the way that they reacted to the characteristics of the data, you can see predominance of one class over the others, but in some cases they lack the ability to have a measurement of precision or to figure out what covariates are actually contributing to that good result. And so that's why you see a mixture rather than one. And we're putting together, and we've got a description of the attributes of the models as well as how they work. And there may be other particular models within these classes that also may be advantageous that weren't included in this particular evaluation. But no, we haven't found a silver bullet.
Okay, thanks. One more
So
question.
you'd recommend that regulatory body would call for all of these tests?
I'm sorry, I didn't quite...
So you would recommend that a regulatory body would call for all of these tests?
No, absolutely not. What I would recommend, the strongest recommendations I make is that there would be both surface and bottom surveys during the initial characterization, which would enable you to hopefully figure out what is dominating the site that you're looking at. It also enables you to get those scaling factors if you want to do a site domain. And then to think about, given the knowledge of what's out there, and often with these places being very, very dynamic environments with current speeds exceeding 2 or 3 meters per second. I mean, they haven't had a long history of sampling at all. You can't fish in there. To think through and choose your indicators, having chosen those indicators, knowing the characteristics, then that should narrow down your choices of models very, very quickly. So we're trying to make it much more efficient and lead towards a standard approach at this point. No, I would not impose this on anybody. And the grad student that walked in the door, she didn't know either.
Okay, just one last question from you.
It's really interesting. It's really interesting. The regulatory process that we have at the moment really emphasizes protected species, as you mentioned, mammals and birds. And indeed, in some cases it's direct impact. In some cases we're worried about maybe displacement away from feeding ground, that sort of thing. So the key thing is the impact on not on the individual within those populations, but on the population. That's what we're required to do through Habitats Directive, Birds Directive. So I'm trying to work out how I can make the link between your stuff in the water column and the things that I'm writing licenses on, which is nice cuddly things like birds and seals.
Okay. I've started using acoustics as my sensor. That comes from something that we play with. I don't feel that this approach is limited to any type of sensor. That's the first point. The second point, even though I use the generic term stuff, there are tools and samples and knowledge that you can use to classify the stuff into categories so that they're maybe identified, which then may identify those species of special status. Similarly, we don't have to look at the whole water column. We can slice and dice it any way you like. And it wasn't until literally last Friday that I think I've got a new set of the next step to give you an approach of which would connect your particular species of interest to look at what processes are important. Sorry, I can say processes here again. Processes are important, and therefore choose the appropriate indicators and metrics that you're tracking the variable of interest. And that, I think, is not what the community, this community, has spent any time thinking about at all, of actually how do we enable the viable monitoring of population impacts as opposed to those particular impacts.
Okay.
Okay.
If there are no more questions, I'd like to thank John again. It was a very illuminating talk, and thank you very much for coming on today.